name is Jim Handa. I am a physician scientist from the Wilmer Institute, Johns Hopkins in Baltimore, Maryland. I study age-related macular degeneration as well as see patients who have macular degeneration. So my week is complex. It starts out by seeing patients. I operate on patients as well at the beginning of the week, and then the end of the week I spend time in my laboratory. So a lot of the research ideas which we've developed for studying has been really come from listening to our patients. So this particular project I'm going to talk about was really kind of derived from patients who come in with macular degeneration, and they oftentimes come in as a second opinion. They have 20/20 vision, and their previous doctor will say they're doing great, and they feel they're going blind. And if you actually listen to their complaints, they feel like they can't get enough light into their eyes. They have trouble adapting to different lighting conditions. One of the most common complaints that I hear is when a patient isn't driving from outside into a parking garage. The darkness once they go into the parking garage is disorienting. They can't see well until their eyes adjust. So that prompted us to think about some of the problems that patients get, and it really comes down to the sensitivity of the retina detecting light is reduced. And when we started to think why that happens, our interest is in the cell type called the retinal pigment epithelium, which supports the health of the retina, in particular the photoreceptors. And we and others have found that the mitochondria, which are the powerhouse of the cells, becomes impaired as we get older, or as I tell my patients, as we collect birthdays. And with the reduction in the ability of the mitochondria to function, they're the powerhouse of the cell. They make the energy for the cell. The cell undergoes a lot of detrimental changes. And one of the things we found almost through serendipity was when we set up an experimental system to impair the mitochondria, the cells, the RPE cells, change shape. And the shape change was reminiscent of a condition or a program called epithelial mesenchymal transition, or EMT. And EMT is a process where a cell is under a lot of stress, and it compensates by trying to survive this harsh environment, but it loses some of its specialized functions. And one of the specialized functions it can lose is in the RPE cells, something called the visual cycle system. And the visual cycle system is designed to regenerate some molecules that are very important in allowing vision to go. And what we've shown is that when the mitochondria become impaired, the cell shape changes, it goes into this program called EMT, and we have a number of different markers that identify this particular type of process. And we can see that some of the visual cycle proteins are becoming impaired. So that whole process could mean that it's a way for the photoreceptors of the retina to become less sensitive to light, and it could explain why the patients have some of this decreased retinal sensitivity. They may have 20/20 vision but still have these visual complaints. And part of the reason for that is if you understand the anatomy of the macula, the centermost area of the macula contains the cone photoreceptors, and they're involved in being able to see 20/20 vision, recognize faces, color recognition. But around that area, we call the parafovea, right outside of that, is a very rod photoreceptor intensive area. And rods are more important in adapting to light, movement, those types of things, but not the acuity. And work from a number of people have shown that the photoreceptors become impaired or die in that parafoveal region first. And to try to explain that, we can actually see some of these abnormal changes to the RPE before that, in that same region. So our working theory at this point is the impaired RPE changes from the mitochondria failing early on explain some of the shape changes that we see when we look under the microscope. to these cell types, but the abnormal function of the RPE makes it so that the interaction and the rod photoreceptors become damaged, and that can explain these early changes. Yet, the photoreceptors in the center of the cones are still perfectly preserved. So while someone has 20/20 vision, they could be perceived as having perfect vision, but because the parafoveal rods, which are important in retinal sensitivity, are damaged, our theory, our work in the laboratory is providing perhaps an explanation for why they have these funny symptoms that where they feel like they really are losing a lot of vision. Well, most of the work we've done has been in cell culture, so we look at cells in isolation, and we have identified the pathway where we think the cell goes into this abnormal shape. So for instance, the mitochondrial damage creates oxidative stress, and that activates a molecule called NRF2, which is a transcription factor that causes the production of a lot of different proteins which protect the health of the eye. That causes some other signal transduction pathways that activate that eventually will cause the transcription or the production of some of the genes that are involved in this process of EMT. So we can show that in cells in isolation, but the next step is we have some genetically modified mice where we can disrupt each of those pathways and see if we can recapitulate the changes that we see in a dish in the animal in a much more complex environment. And if we can show that those changes mimic some of the features of macular degeneration, then we think we have a great model in a live animal. And then there are, from the cancer world, there are different molecules that can reverse this process. At least it's been used in cancer, and we can try to apply some of these different molecules that will reverse the EMT process. And if we can restore the normal health of the RPE cell, that might be a way to preserve or reverse and improve the health of the overlying retina. So I think it would be, if we can show it in our mouse models, the next step would be, depending on the different molecules that we could identify, the next step would be to understand, there's two potential pathways. One is, can we give this medicine through a pill form, and we can test that to some extent in the mouse models. If it's more local therapy, we would need a larger, more relevant sized eyeball, such as the pig or such as the rabbit, where we could do more pharmacokinetic studies to look at the distribution. On the other hand, we can also think about working with some of our nanomedicine colleagues to provide more sustained release formulations, because some of these different strategies can have anywhere from 3 to 6 months of sustained release. So if it requires an injection into the eye, in patients who have wet macular degeneration, everybody knows that they get as frequently as monthly injections into the eye. This strategy potentially would just be once or twice a year, and that's a much more palatable treatment approach for our patients.